Okay, let's get started. We're gonna talk about special senses today. We're actually coming toward the end of lecture exam pre-material. Very first week of class, I think it was in lab, I talked about the lab schedule. And how at the end of the semester, we have lab exams every other lab. You guys remember that? We are entering that time. I said that there were going to be 3 weeks in hell for you because lab exams are every other week. But it's going to be lab exam, lecture exam, lab exam. You guys remember that? That's our time right now. So this is what the calendar looks like. Here we are today. Next week is lab exam 3. Following week is lecture exam 3. The following week after that is lab exam 4. This is where you're going to have to really organize your time, okay? Oh, the DD is drop deadline, right here. My goal is to have lecture exam 3 graded and posted probably by Friday morning at the latest. So those that have to make a decision based on lecture exam 3 can make it with information. Okay? Any questions? All right, let's talk about vision first and the eyes. Some of this we covered in lab, so this should be reinforcing. This is telling you what you need to know for lecture exam questions. For example, you do need to know the 3 layers, coats, or tunics. The outer one is a complete layer. That's a fibrous tunic, or scleroid coat is the other name. It's made up of 2 things. The white of the eye we call the sclera, and then the clear anterior portion of this tunic, which is the cornea. Again, both is made up of collagen. That's just how the collagen is arranged to make an opaque white or clear. The cornea does not have conjunctiva on it. It just has a very delicate, thin stratified squamous epithelium. It's not like our skin, our epidermis, where you have multiple layers. It's just a few layers. On the inside is simple squamous, very, very delicate. Okay, everybody got this? That's the outer fibrous tunic. Again, A plus B equals C. The conjunctiva is actually a mucous membrane. And as a mucous membrane, it has to have tissue type 1 and 2. It has to have epithelial tissue and connective tissue. Okay? And in that connective tissue are these blood vessels. That's why the white of the eye has these blood vessels that you can dilate if you have bloodshot eyes. Everybody got it? You do not want blood vessels over your cornea. It's going to get in the way of vision. That's why the cornea does not have conjunctiva. No blood vessels, just epithelium. Everybody got it? Do I need to repeat that?
Yes.
Conjunctiva is on the anterior sclera. And that's what these blood vessels are showing you. Conjunctiva has mucous membrane. It is a mucous membrane, so it has epithelial tissue and connective tissue. 2 types of tissue. In tissue type number 2, connective tissue, are blood vessels. Those are the blood vessels you see in that picture. Those are the blood vessels that dilate when you have bloodshot eyes. It is the blood vessels in conjunctiva. Are we clear? The cornea does not have conjunctiva. It ends about at the border between sclera and conjunctiva, because you do not want to be trying to see through blood. There is no blood vessels on top of your cornea. Are we clear? So if there's no blood vessels on top of the cornea, what keeps this alive? It delicate shred, that's living cells. They don't have blood supply. How are they getting what they need? I'll just tell you. I got a feeling you're not gonna guess, so I'm just gonna tell you. In tears, there's enough nutrient supplies to keep the cells alive. They are not highly metabolic, so they don't require a lot of nutrients. And the oxygen they get is the oxygen that is in the room air. They don't have to get it delivered by red blood cells. They get the oxygen from the same air you're breathing. Everybody got it? Does anybody here wear contact lenses? Back in the day, when the contact lenses were hard, one of the major advances in the contact lenses was, maybe you've heard this term, gas permeable contact lenses. Have you heard that term?
No.
That is why, if you're gonna cover the cornea, if that contact lens is not gas permeable, it doesn't have any way to get the oxygen. And in the old days, that's why you can only wear contact lenses for a really short period of time. Or you're gonna choke off these cells. Does this make sense? Does that help to tie things with, everyday stuff? All right, the middle tunic is the choroid coat or vascular tunic. It is also a complete layer. Posterior to the ora serrata, we learned in lab, is just choroid. That's it. Anterior to the ora serrata is where things get complex. The ciliary body starts the anterior side of the ora serrata. It has a ciliary muscle that changes the shape of the lens. It goes all the way anteriorly to the iris. But the suspensory ligaments and lens is also part of this choroid coat. Okay, the iris again has a pigmented cell layer. You guys ever do that thing in grade school where they try to teach you about Mendelian genetics? Talk about eye color. They never seem to really work out. That's because versus, like say, pea plant flower color is single gene. But both hair and iris color is controlled by multiple genes, each with multiple alleles. It's not going to be as clear cut as pea plant flowers. If you ever did that in grade school and it just didn't work out, it's because it's more complicated than Mendelian genetics. But here you can see the blue layer is the choroid coat. It is considered a complete coat. And in red is the retina, and you can see the inner tunic is partial. It only goes up to the ora serrata. And again, the iris controls the size of the pupil. The diameter, depending on how much light there is. So here outside the sunlight, the constrictors make it overly tiny. When you come into this room, it opens up a little bit bigger. If I close the doors and turn off all the lights, it gets really big. Everybody get you can't control it, it's smooth muscle. All right, the inner coat is the retina or neural tunic. It has multiple layers within it. And we're going to look at that here in a second. But the outer pigmented layer is what is up against the choroid. It is the most outer layer of this retina. And again, this retina extends anteriorly only as far as the ora serrata. So, up against the outer pigmented layer, so maybe I should add that over. We have the photoreceptors. Now I drew a rod here, or it could be a cone. But they're right up against the outer pigmented layer, pointed away from the lens, as we talked about in lab. We'll get to the cells of the retina in a second. By the way, this is one of the things that would be good to know going into physiology, cells of the retina. But if we look at a macro view of the retina, especially if we're looking at through the cornea with the scope, we would see that in the visual axis is a yellow spot, and it's called the macula lutea. So this is in the visual axis, center of the cornea, center of the pupil, center of the lens. Draw a straight line connecting those dots. You're right in the center of the macula lutea. But right in the center of the macula lutea is the fovea centralis. There you have only cones, you don't have rods, and they're really packed together. Really densely concentrated. And, like we talked about in lab, it's a divot because all the superficial covering layers are all shoved off to the side, 360 degrees around, and that's what forms the divot you see on the screen. So not only do you have the highest concentration of cones, they are bare cones. All the superficial layers have been shoved off to the side. And then to the medial of the macula lutea, and the macula lutea is right here. Fovea centralis is right in the center of it. To the medial of it is where you see the optic disc. And that's where all these axons are exiting the eyeball. So in that way, the blind spot can be considered the opposite of the fovea centralis. Fovea centralis is all photoreceptors, in this case cones. No fibers. That's why it's a divot. Fibers have been pushed off to the side. The optic disc is all fibers, no photoreceptors. Do you see how it is, in a way, like the opposite? Fovea centralis, all cones, no fibers. Optic disc, all fibers, no photoreceptors.
So like with the fovea centralis, it's pushing all the fibers away, is the disc pushing the photoreceptors away?
They're all gathering. All the fibers are gathering at the disc.
Fovea centralis pushing away and they're gathering at the
Uh-huh.
disc. Blocking any cones or rods from moving.
Yeah, there's just no room to have anything other than fibers at the optic disc. Did everybody follow what I just said? Because it's a way of understanding the retina. And that's why I don't have it memorized. And we did this thing here with, in lab with binder paper. Wasn't that freaky when we noticed the blue line went right straight through even though the dot disappeared? This would be, if you were using this, you would be using your left eye because again, the blind spot is inverted, so it's lateral. So even though the optic disc is medial, this would be a left retina if we were looking at a left retina. So even though the optic disc is medial, the blind spot is lateral. So this would be for a lateral left eye. And if you make the dot disappear, these diagonal lines will all line up and be continuous to the blind spot. Isn't that weird? But there are limitations to this though. Like, okay. If this was what was going through in the blind spot, and this was where the dot was that disappear, your brain would actually make the two horizontal lines on the same plane, even though they're not. You have to open up both eyes to see, oh no, wait, they're on different planes. With one eye, with the blind spot, it looks like it's a straight level line. But do this, your brain will not align up vertical lines. And so in the blind spot, even though the dot disappears, all you see is kind of like a gray. You actually don't see an intact line. So there are limitations to what your brain can do to fill in the illusion. It's not limitless. Okay, this room is too light, this never works, but one of the things is that we talked about the superior colliculus as being, your visual reflexes, which is why it's actually hard to focus and stay focused on that plus sign. Because your superior colliculus doesn't like things staying in your blind spot. And you'll just flick your eye a little bit. And that's all you gotta do is flick your eye a little bit, and that dot never disappears. And you won't even notice your eyeball's moving. You think you're focused on the plus sign, but your superior colliculus is just doing this little subtle flick, so that dot never disappears. That's why if you couldn't get the dot to disappear, that's because you don't have command over your superior colliculi. It happens. There's not everyone can make the dot disappear. It is a discipline thing. Well, same thing goes with this. Cones need a lot more light to activate. Rods, you can see in the dark with rods. But since rods doesn't give you any information about color, at night you see in grayscale. Does everybody understand that?
Yeah.
You look down the street, unless something has its own inherent color, like brake lights or certain kinds of neon signs, you do not see in color at night. Look down that street, there could be a bunch of parked cars of different colors, they all look just different shades of gray. Because only your rods can activate in that low light. Cones need more light. Well, what's in the fovea centralis? Only cones. If I could get this room really dark and have these little stars come on the screen really dimly, your superior colliculi will not let you focus on these dim stars because the cones cannot activate in the fovea centralis. And it's the weirdest thing. You'd be trying to stare at a star and pretty soon you're not. Superior colliculi, visual reflexes. All right, you can try that at home with your computer, especially if you could dim the screen. Just try to focus on the dim little star. Keep it in the fovea centralis and your superior colliculi will not allow it. All right. The lens is also part of the choroid coat, so next to this slide, right, choroid coat. It is held in the visual axis by suspensory ligaments. On one end of the suspensory ligament is the ciliary muscle. On the other end of the suspensory ligament is the equator of the lens. Do I need to repeat that? On one end of the suspensory ligament is the ciliary muscle, which is part of the ciliary body. On the other end of the suspensory ligament is the equator of the lens. The suspensory ligaments and the lens form the boundary between the anterior compartment and the posterior compartment. But in the slide, I couldn't fit the word compartment in. The other word for compartment is cavity. And you just gotta know that. But I like to use the word compartment because it's bigger. And the compartments are the bigger spaces. Here you can see the anterior and posterior chambers are part of the anterior compartment. Everybody got it? And behind the lens and suspensory ligament is the posterior compartment. Filling the space in the posterior compartment is 2 bags of jelly. The jelly is called vitreous humor. The 2 bags give your eyeball its shape, which if it is misshaped, could make you either farsighted or nearsighted.
When you say the equator of the lens, are you just talking like, say, center of it?
Well, this week in lab, we're gonna do eye dissections, and when you pull the lens out, it's like a marble. The lens naturally is more spherical. So if you think of it like a globe, right at the equator, okay, they do this to be a little bit flatter here. But if this was like a ball, that would be the equator, and it's forming a ring of attachments. Okay?
So it's not just right there, it's all the way.
That's all the way around. But it's only on the equator.
Got it.
Okay? It's the only place you have the attachments of the system. Let's talk about eyelids, or palpebrae. They work like windshield wipers. They are not just skin. There is a plate of connective tissue, this broad sheet that gives your eyelid some stiffness. The skin is really flimsy all by itself. You have an upper tarsal plate and you have an inferior tarsal plate. The only one you can consciously move is the superior one. You cannot move the inferior one. Laying on top of the plates are these modified sebaceous glands called tarsal glands. Used to be called glands of Zeiss. I'm so glad we're getting rid of all those proper names. Everybody got these? Now, a lot of these glands are doing kind of the same thing. They're working together to keep the windshield wipers from sticking together and lubricating it so they can slide easily over the eyes. Tarsal glands, again, are modified sebaceous glands. Some of them will empty up on eyelashes, but a lot of them don't. So like sebaceous glands are all empty out in hair follicles, you guys remember that? Some of these do on the eyelashes, but not all of them. And then on the medial side, right next to the medial palpebral canthus or commissure, is a lacrimal caruncle. We saw that in lab. And it is secreting a lipid-rich secretion, which you might see that secretion in the morning. Hi, ghost, sleepies. In Japanese, it's Namek, so.
Oh,
I actually used to speak Japanese back in the day, but my parents couldn't speak it in the house, so it all disappeared. But Japanese is a hilarious language because it's like 4 levels of formality. You know, there's the top level, which you'd only speak to dignitaries and the emperor, and then other ones where you would speak to your teacher, which was respectful. And Japanese are really respectful of teachers. Here, you know, they just rip you a new one on Rate My Professor. Who gives a... All right, anyway. And then there's one you talk with, like, with your coworkers and, you know, family that maybe don't live in your house, but it's more informal. And then down at the bottom is for farm workers, which my family were sharecroppers, and restaurant workers. It's very full of slang. But the words all make sense, like meme is I, kuso is shit. Mimi is nose. If you got a booger, that's Mimi kuso. Okay. Again, all these glands are working together to keep things lubricated so the eyelids can perform their windshield wiper function. Now, again, the tarsal glands are modified sebaceous glands, And when sebaceous glands get infected, that's a zit. That's basically what a sty is. A sty is a zit, but it's not an irregular sebaceous gland. It's in one of these tarsal glands. And because they are so big, these can get incredibly swollen with a ton of bacteria. Okay? And you don't want this much bacteria pouring into your eye. You just don't. So never, ever, ever pop a sty. You will probably have to go to the ER at some point if you pop a sty. You know what you're supposed to do if you get a sty? Hot compress. Basically what you're doing is you're increasing the circulation to the eyelid so that it drains internally. And your body's own defenses will take out the bacteria internally and not infect your eye. Has everybody got it? Lovely. All right, let's talk about the conjunctiva. Again, it is a mucous membrane, so it has stratified squamous and connective tissue, loose connective tissue that has the blood vessels. There are two subforms of conjunctiva. One is on the inside of the eyelid. The other is on top of the sclera. Palpebral conjunctiva is on the inside of the eyelid. Bulbar conjunctiva is on the sclera. These are the blood vessels you can see when someone has bloodshot eyes. Those are the blood vessels of the bulbar conjunctiva. So it's all there in yellow. And on the cornea, there is no yellow. Everybody see that? There's a lot more to the anatomy of the eye than what's on this PowerPoint. I'm just telling you what you're responsible for lecture 10. Okay? But don't think what I'm teaching you here is an exhaustive list. This is highly abbreviated. Okay? This is the cornea with epithelium, not conjunctiva. So again, no conjunctiva on the cornea, only on the sclera. All right, we covered this in lab as well, the lacrimal apparatus. Remember that the lacrimal gland is on the superior lateral corner of the orbits. You got about a dozen ducts that empty tears on the lateral side of the eyeball. There's actually plenty of things in tears. I've listed one enzyme called lysozyme. It tends to punch holes in bacteria, so it helps to fight. It's a non-specific way of fighting the surface bacteria on your eyeball. It also has IgA, which is an immunoglobulin. It has other things. Even though it's mostly water, it has a ton of things, including enough nutrients to keep the corneal epithelium alive. Yes?
So it'll keep the nutrients going even if you don't cry?
Yes, because basically what crying is just excess tears. There's always tears 24/7 on your eyes. Crying is just excess tears. So this flow is actually going 24/7. It's just, if you're not crying, you don't notice it because it evaporates before it even gets to your nose. Everybody got it? The only time your nose runs is when you've got an excess of tears draining out of your lacrimal sac, because that's on the other side. So the sequence, go ahead and fill in the blank and then I'll tell you the sequence.
Does that ever get backed up? Like, you know, you get skies from the sebaceous glands in your eyelids, if that gets clogged, or is it in that?
Yeah, you know, the only problem you would have, and it would be extremely rare, is for all a dozen ducts to get plugged.
Oh, it's not just one big one?
No, there's 12 different, 10 to 12, that's what it says there, 10 to 12 different ducts empties the tears from the lacrimal gland. You'd have to block all of those. If you don't do that, it's not gonna be a problem, because again, normally unless you're crying, you don't have enough volume for it to back up anywhere. Okay, and even if you did have a backup of the nasal lacrimal gland, all it will mean is that instead of your nose running, your tears will just run on your face.
Oh, okay.
Okay, everybody want the sequence? Lacrimal gland, 10 to 12 ducts, lateral side of the eye, goes from lateral to media, the 2 lacrimal canaliculi, then the lacrimal sac, then the nasolacrimal duct that takes it, and it takes it all the way down to the bottom of your nasal cavity, so that's kind of nice, instead of going from the top all the way down. It actually, the nasolacrimal duct empties on the inferior meatus, which is underneath the inferior nasal concha. Everybody got it? Everybody got the sequence? Do we need to say it again?
Yes.
Lacrimal gland, 10 to 12 ducts, lateral side of the eye. Lateral to medial. 2 lacrimal canaliculi. Lacrimal sac. Nasolacrimal duct. To the nasal cavity. It says there it's not highlighted, underlined, or bolded, but it's the inferior meatus of the nasal cavity. Okay.
Yeah.
All right, this slide may be a small essay for lecture exam 3. I need for you to pay attention and engage, because what I'm about to say is very simple, but it'll go past your ears if you're not engaged. There are 2 potential short essays on here, and both are a set of 4. So there's 2 sets of 4. You've got to read the question and answer my question. I just gave back yesterday physiology lecture exam that they took last Wednesday. There were 2 potentially large essays on that. I give them a handout. You guys are not even given a handout. I give a handout of potential essays with the content on them. Okay, they gotta put it in complete sentences, but I give them the content, right? The question is right there, and then boom, the content. The question is right there, and then boom, the content. This one only had 4 essays on it, of which I asked 3. You know how many potential essays there are in the physiology lecture exam? In Questa? Upwards of 30. I gave 4, of which I asked 3. These people think I'm a dick. And I have to tell them, no, my job is to make sure you're a grown-up and you know the real world. And here's the real world. I gave you 4, they gotta do 30. So quit your bitching and just do it. Oh, he's a dick. No, you gotta compete with those people. How are you gonna do that if you think things are supposed to be easy down to your level? Well, anyway, the 2 of the essays are large, 2 are short, and I had at least 4 students answer the other long essay, even though the question is copy and pasted from the handout. There is no way to even misinterpret which one I'm asking, because it's copied and pasted from their handout. And they gotta compete against those people who are doing 30 essays. So am I a dick for saying you better get in the ballpark? Only a dick would not get them into the ballpark. Would perpetuate the lie that you're in the ballpark when you're not. That's a dick. Well, I should probably change my language since we're not covering reproduction till the end. Do you hear what I'm saying? And so a student asked me, well, can I get some credit because I did the other essay? I'm gonna tell you what all anatomy teachers should tell all anatomy students. Answer my question. If you change it in your head to your question, you can get your question right and have 0 points for it. Because you did not answer my question. This is a lesson that will help you for any entrance or licensing exam. Any. Answer the question as it's asked on the page. Especially when you take those tests, because you don't even know the guy who wrote it. You've got to be able to read with comprehension. And that means answer the question that's on the page. Oh, you get it? There's 2 sets of 4 on the slide. You have got to recognize the words of the question because it's clear cut, clear as day. There are 4 steps to focusing on an image. They are convergence, and these are the extrinsic eye muscles. To focus on something, your eyes are actually not pointed in the same direction. They are not parallel. They are converging. If it's something way distant, they look like they're parallel, they're not. They're still converging on a spot that's very distant. But where you can really notice it is when that focus gets closer and closer. That's convergence. But if it's distance, you can't tell that they're actually still converging on something. Everybody got it? They look parallel, they're not. Second, you have to have the right amount of light hitting your retina. This is what your iris is supposed to do with the constrictors and dilators. Again, right now, your pupil's medium size. I turn off the light, it gets really large. You go outside, it gets really small. That's the second step. This is an old film camera. They would actually call this the iris on the camera. It's doing the same thing. Accommodation, this is actually changing the shape of the lens. I can explain this to you if you want, but basically this is the third step. The ciliary muscle, since it's in a ring, the ciliary body is shaped like a ring. The ciliary muscle is a sphincter muscle. Okay. But you gotta focus on something far away, you relax the ciliary muscle, and the opening gets bigger, that makes the lens flatter. If you wanna focus on something up close, you have to contract the ciliary muscle to make the lens rounder. Everybody got it? But this is why, okay, I'll just explain this to you. You don't have to write this down. Sphincter muscles, like the ciliary muscles, are counterintuitive, right? Because like with the biceps brachii, you contract it, all the ligaments and tendons get tight, correct? Because the ciliary muscle is a sphincter muscle. It's shaped in a circle. And to close the hole, you have to contract it. Let's hope everyone's sphincters downstairs are fully contracted. So this is what the ciliary muscle look like if it's relaxed. This is what the ciliary muscle look like if it's contracted. Well, if you're watching a football game way from the stands, you want the lens to be flat and not bend the light so much. And this is how this is counterintuitive. Because it's a sphincter muscle, when it's relaxed, it opens. But if it opens, the suspensory ligaments tighten up, and that flattens out the lens. This is how it's counterintuitive, or the opposite of the biceps brachii. You contract the biceps brachii, all the ligaments and tendons get tight. If you want to get the suspensory ligaments to be tight, you got to actually relax the ciliary muscle, not contract it. Do you see how it's the opposite? If you contract the ciliary muscle, you make the lens round. And when you contract it, you're closing it, that makes all these guys loose. It's the opposite of the biceps brachii. Well, convert this to the experience of being human. You can watch a football game for 3 hours, and your eyes aren't tired, because the ciliary muscle is relaxed for 3 hours. You study for lecture exam 3 for 3 hours, with your notes and your screens or your handouts or whatever, and your eyes are tired, because the ciliary muscle is contracted for 3 hours. Convert it to the experience of being human. Anyway, that's step number 3. I wasn't even going to go into that. Here's the 4th step, which you will see is another set of 4. You have to bend light to focus it on the fovea centralis. And the 4 refracting media in order are, and they're in parentheses, cornea, aqueous humor, lens, and the vitreous humor. The lens is the only one that you could regularly change. The other 3, you can't. Well, you can change the cornea once or twice. How do you readjust the cornea?
What is refraction?
Light bending. How do you change? You change the lens readily. Just look distance, look close. Look distance, look close. You're changing the lens. How do you change the cornea? LASIK. That's what they're doing with LASIK, is they're changing the shape of the cornea, but you can only do that once or twice. The lens is the only one you can change readily. Any questions? So the whole slide is 4 steps of focusing an image on the retina. Number 4 in this 4 steps is the 4 refracting media in order. There are 2 different sets of 4. Answer my question. Four steps of focusing image on the retina is the whole slide. The 4 refracting media in order are right here in the parentheses. Answer my question. I'm just glad I'm not giving you 30 possible essays for lecture exam 3.
I guess I'm struggling to understand how that last one, I can understand how to focus on an image as a question, but the bottom one, like, how would you word that as a question?
what are the four refracting media in order?
Okay, cool. Just wanna make sure it's not like.
I mean, I'll make it as clear as I can, as I strive to teach defensively.
Thank you.
Well, yeah, like with physiology, I mean, I copied and pasted the question from the handout. I don't know what else to do about that. If you answer the wrong question, be aware of it for next time, and then you can get into the ballpark. All right, so this is just evidence of how I'm not a dick. This is what I told her, because she actually is a good student, this one that actually came and asked me, can I get some, I would've gotten an A, and she got a B, even with that. Could've gotten an A. I said, listen, I'm not gonna change your grade because I want you to feel the sting. And the sting will maybe make you read the questions more carefully. But then I said, if you are borderline at the end of the semester, bring the blue book and let's talk. Because that amount of points will only make a difference if she's right on the board. If she's in the middle, it's not gonna make a difference. So just, all right, we'll talk if I have to talk about it. I'm not a dick. But I'm not changing her grade because she's gotta feel the sting. It's the sting that makes you more careful about reading test questions. Does that make sense?
Yes.
All right, let's talk about the photoreceptors. Photoreceptors at some point have to have a chemical that is affected by light. Okay, and the chemical that is affected by light is this chemical called retinal, also referred to as retinine. It could be either for all purposes. It's organic chemistry difference, don't worry about it. But it's a fatty acid. Remember the phospholipid bilayer, the two long tails?
Yeah.
One of those tails is a fatty acid. So if you have this long chain fatty acid, it came from vitamin A, which is a fatty acid. It's a fat soluble vitamin. And then you have the two, well, let me just show you this. This is the form of this chemical in the dark. And it's got this double bond of the 11 carbon, that's why it's called 11-cis retinine or retinal, and it's bent at the 11 carbon, and it sits on top of this protein called scotopsin. Remember protein boulders? It's like a protein boulder. And when you're in the dark, this 11-cis retinal hugs this boulder because it's bent. Everybody got it? Then when light hits it, it changes to this trans-all or all-trans retinal. And when you break that double bond, it straightens out. And it partially separates from the boulder. That's what starts the chain reaction of vision. Dark, light. Relaxed form, excited form. And I tell my physiology students, use whatever metaphor you want for it getting excited. And A plus B for rods equals C. Rhodopsin is the name for the photopigment in rods, and it's made up of scotopsin and the retinal. A plus B equals rhodopsin.
So when you say A plus B, it's the A over the B.
Retinal plus scotopsin equals rhodopsin.
But what I'm saying is only when it's like this.
It doesn't matter.
Okay.
Rhodopsin is in either form.
Okay.
Okay. Again, so rods is what has rhodopsin. It's much more light sensitive. You need a lot less light to activate rods. But it doesn't have really good acuity because you don't have rods in the fovea centralis. You only use rods for peripheral vision. Does not distinguish between colors. I would circle black and white, draw a line off to the side, and write grayscale. It's really not black and white, it's how much gray. Cones will give you clearer images, especially since they're found in the fovea centralis, but you need adequate light. And they're going to give you color perception from the 3 different types of cones that you can see here, blue, green, and red. True or false? To find that right color paint at the paint store, you only need cones.
Say that again?
To find the right color paint at the paint store, you only need cones. It's false. In fact, most questions where you see always or only or never is usually false. That was in the test taking strategy. Because here's the deal, has anybody ever gone shopping for paint?
Yes.
All right, well, if you have never done shopping for paint, you're not missing anything, but I'm gonna tell you what it is. First of all, you have to find the right strip, right? To find the right strip of paints, you only need cones, because that's just about the color. Have you ever seen a strip of paint? If you look at the different colors or the different tabs on that strip, it is the same color, but it has different levels of gray. What artists call value, it's grayscale. So to find the right strip, you only need cones, but to find the right tab on that strip, you need rods and cones, because rods are giving you information about grayscale. Does that make sense?
Sure, thank you.
Oh, the sounds Japanese, okay. Everyone can see the 1 and the 2, correct? But people who have red-green color blindness may not be able to, don't yell them out. Now, you can either have a problem with green or you can have a problem with red. Well, now you know there's a 9 on this right one. And if you can only see the 9, you have a problem with chlorelade, which is the protein for green cones. If you only see the 6, now you know the other number, you have a problem with erythrolate, which is the protein in red cones. Does anybody here see both numbers on the left? Does anybody not see both numbers on the left? So everyone sees both numbers on both of these? Okay.
Is it 1 and 6 on the ones?
Yeah, 1 and 6, 9 and 6.
I don't know what this comes from.
I've had a handful of students not be able to see all the numbers here. And so far, they have all been male. Okay, so when you talk about protein synthesis, and again, this is a problem with chlorelate and erythrolate, which are proteins. You'll learn in physiology is that they're all coded for on your DNA. DNA is your chromosomes. In the case of these 2 proteins, those genes are found on the X chromosome. In other words, they are sex-linked, specifically X-linked. That's why most of the students that have had trouble with this slide have been male, because it's a recessive trait. And let me explain that really quick. I'll do it over here on this corner because it's really not that. In biology, we do this thing called the Punnett square. And you might have done this when you were doing grade school Mendelian genetics with pea plants. But our cells are diploid. You guys remember that with meiosis? We have 2 of each. Well, we have 23 pairs total. The first 22 pairs are called autosomes. The last 23rd pair are called the sex chromosomes. And females are truly homologous XX, and males are XY, even though the Y is very dissimilar to the X in meiosis. They actually do act as homologs, okay? So they're still considered homologous even though they're very similar. So what you do in the Punnett square is you do the meiosis in a table. You split the homologs. That's what happens in meiosis 1. Does this sound familiar at all?
Yeah.
So here's mom, here's dad. They bump uglies, and all you gotta do is match the columns and the rows. Everybody see that?
Yeah.
This should tell you that, especially when it comes to births, our species is approximately 1 male to 1 female, which it is. When you talk about live births, it's about right down the middle, 50-50. Can you see that in the Punnett square?
Yes.
How come there's a lot more females than males? It's good for males. Why are there more females than males? Nobody? Because go ahead. Testosterone makes you stupid. Man, if there's a fight outside of Elmer's and somebody dies, that's a dude. Right?
Yeah.
Driving statistics. Fatalities involve more males. Right? Yeah, you just gotta watch like MTV Ridiculousness. All the stupid stunts, it's a dude. And if it is a chick, there's a dude behind her going, oh, it'd be really cool if you did this. We just kill ourselves. And that's why there are more females than males. But by birth, it is 1 to 1. Are we clear? All right, well, these chromosomes that cause color blindness are on the X chromosome, and it's recessive. So let's say mom has the bad gene, but because she has a good gene that shows dominance over the bad gene, she can see red and green just fine. Let's say dad can also see red and green, but he may not dress like it, but he can. Then you gotta just match up the columns and the rows again. So in this column, everybody has good genes. On this column, this daughter's like mom, what we call a carrier. She's carrying the bad gene, but she doesn't show any problems from it because she has a dominant normal gene. Everybody got it? But because it's on the X chromosome, this poor son doesn't have another X to show dominance over the bad gene. If he gets the bad gene, he's stuck with it. That's why when it comes to what are called X-linked recessive traits, which this is, on the X chromosome, and it's recessive, it tends to be in males. This includes red-green color blindness, includes hemophilia A, which is in the royal family. So if you look up the royal family's pedigree and hemophilia A, you will see it skips some generations. Sometimes it's in the same generation or in the next generation, but most of the time it's skipping because it's recessive. And the people that have hemophilia A are male. What's that? That's where you have clotting difficulties, and you can't, and there's multiple ways to having it. That's what's called hemophilia A. And you just bleed to death. Any other questions? Last in the spring in physiology, I had my first student. I've been doing this for 15 years. First female student who was colorblind. So it is possible. It just happens more in males when it's like this, right? For example, dad has to be colorblind. If this is a small r, this daughter is colorblind. Everybody see that?
Yeah.
Mom has to be a carrier, dad has to be colorblind. You can have a daughter, and I asked her, so was your dad colorblind? She goes, yeah, he's colorblind. So her mom was a carrier, her dad was colorblind, she got both small R's.
I have a question.
Yes.
I actually watched this video on Instagram this weekend where a guy was colorblind and they gifted him these glasses and then he could see.
Yeah, and you basically just torque them with the wavelengths. And because like we used to think that other mammals don't see in color, they do, it's just different from our color perception. And you can actually have glasses you can give your dog that can see fully in color like we do. So. What else does a Punnett square tell you? Okay? Since you guys are getting educated, you gotta get educated. Because there are still ignorant men blaming their wives for not bearing them sons. In the 21st century, there are still ignorant men blaming their wives for not bearing them sons. Look at the Punnett square. Is that in the control of the female?
No.
Females can only give the X. The gender of the kids is based on the dude. Guy wants to be bad, he's not getting sons. He's got to point his bony finger at his own balls. All right, let's talk about the neural pathway. What's in this bullet here is what I've drawn here. In the retina, the main pathway is rods, bipolar cells, ganglion cells. Yes, there are horizontal cells around here, there's amacrine cells around there, but they're kind of off to the side, doing other processes like lateral inhibition. But the main sequence that will be really helpful for you in physiology, when we talk about rod photochemistry, is photoreceptor, bipolar cell, ganglion cell. Bipolar cell has only two poles coming out of the cell body. This one is the dendrite, the first one is the dendrite, if the signal's going in this direction. Second one is the axon. The ganglion cell is a multipolar neuron, which means it has multiple dendrites and one axon hillock with one axon. What this is saying on this bullet is that the axon of the ganglion cell is cranial nerve 2. All the axons of all the ganglion cells at that eyeball is exiting at that optic disc and forming the optic nerve after it exits. Do I need to repeat that? All the axons from all the ganglion cells of that eyeball gather at the optic disc, then leave in the optic nerve. But they are simply all the axons for all the ganglion cells of that eyeball.
When you say they gather there, they're not synapsing there, they're just
No.
like hanging out.
They're just all these fibers all come together in this one circular spot.
Still just one axon.
Well, they're all the axons from that eyeball. But they are all converging on this one circle, the optic disc, and then leaving in the optic nerve. So in lab, right, you have to know the cranial nerves and you do have to know the optic nerve. That is all the axons of all the ganglion cells of that eyeball.
I think I'm just getting hung up because an axon in my mind is just like one, I'll call it tail, right? So when the bipolar cell is synapsing with those dendrites, it's multiple axons that are synapsing right there.
It's
Or is there
multiple-
just
it's
multiple
It's multiple
bipolar cells?
dendrites.
And multiple, okay.
It's multiple dendrites that are synapsing, because there could be another bipolar cell on another rod, and another bipolar cell on another rod, all coming together on this one ganglion cell, but it only has one axon. Just think of it as a multipolar neuron. Multiple dendrites, one axon.
Right, so you're saying one axon, but then you're telling me there's a bunch of them hanging out.
There's a whole bunch of ganglion cells in one eyeball.
Oh, okay, so it's, for this picture, this is just one, but
This
there's a bunch
is
of
one.
those situations
Yeah.
happening.
Absolutely. Thousands upon thousands of ganglion cells in one eyeball.
Got it.
Okay? Everybody understand this first bullet then, and how it fits the drawing?
Yes.
Remember, this is counterintuitive. The light would actually be going up in this direction. As has got to go through the fibers of the optic nerve, through the ganglion cells, amacrine cells, bipolar cells, horizontal cells, to finally get to the rods and cones, which are pointed away from the lens. Does that sound familiar? There it is there. And there it is there. Rods and cones, bipolar cells, ganglion cells, whose axon is fibers of the optic nerve that are running on the surface of the retina before gathering at the optic disc and leaving the eyeball and the optic nerve.
So like right there, where there's all these rods and cones, is there like a rod to every cone in that spot, not in the like?
No.
Is the ratio is not the same?
Yes, not the same. And in fact, at the periphery of your retina by the oris aorta, it's all rods. At the fovea centralis, it's all cones. And there's a spectrum, like right here, it's 50-50. You get over here, there's more cones. You get over here, there's more rods. It's a spectrum of mixture. Everybody understand that question and the answer? All right, so let's do the neural pathway. And that's what I'm doing on the whiteboard. And I'm gonna, I don't know if I'll get to it today, but I'll do the thing for hearing. So I'll take a picture of that and reproduce that on Thursday. All right, remember some of the fibers cross at the optic chiasm. Is it all right that I do this in abbreviations? Do I have to write? So this is optic nerve, optic chiasm, optic tract. And the optic tract is what enters the brain. The optic nerve has axons from one eye because some fibers cross at the optic chiasm. Optic tracts have fibers from both eyes.
Got it.
Not all of them cross at the optic chiasm, just some of them. Once it enters the brain, there's a split. About a quarter of them go to the superior colliculus of the corpora quadrigemina. That's this abbreviation. And about 3/4 goes to the thalamus. Lateral geniculate body of the thalamus. Remember that the thalamus is the great relay station for sensory. And all your senses synapse there except for the sense of smell, olfaction. This is where vision synapses, lateral geniculate body of the thalamus. Then there's going to be this huge spray toward the posterior called optic radiations. And it's going to hit the furthest posterior portions of your cerebral cortex, called the primary visual cortex of the occipital lobes.
When you say spray, do you mean like a bunch of axons just shooting out
Yes.
the back of your brain?
Yes, and so on this figure it's in blue. Now, this is an abbreviated pathway. Once you get to the superior colliculi, it gets complex in a hurry. It's going to go to several cranial nerve nuclei, and it's going to involve cranial nerves 3, 4, and 6, so that it could help you, because those 3, 4, and 6 are controlling the 6 extrinsic eye muscles. It gets really complicated, so is it okay if we just leave it here for you guys?
Yes.
How many pairs of cranial nerves do we have? Total. How many pairs of cranial nerves do we have? 12. Right? An oval limbus, towering tops, fattest, extremities, there's 12. How many are involved in vision? 3? Oh, the cranial nerves. How many Roman numerals are involved in vision? And the big hint is it's not just cranial nerve 2. That's 4. 1/3 of your cranial nerves are for vision. The signals for vision, so that your brains can generate an illusion that you see me, is cranial nerve 2. But cranial nerves 3, 4, and 6 move your eyeballs. And they're getting sensory input from the superior colliculum. That is why you can track a moving object. Even if you're moving at a different direction and speed, you can still track that car. You're going this way, you can track that car going that way. Because of the fact that 1/3 of your cranial nerves are involved in vision. Everybody got it? So yeah, it gets a lot more complicated than what I have you study. Okay. Pop quiz. That's more for a lab though, isn't it? Okay, let's just do a couple more. Let's go to the ear. Remember on the ear, it's about sets of 3, isn't it? 3's all over the place. So we have external ear, middle ear, and inner ear. In the external ear, you have the auricle, which is elastic cartilage, generally beat up by piercings. Does anybody here know what that is?
What?
It's a keloid. Yeah, it's not an earring. Well, I guess it is. It's a collagen flesh earring. Keloid, so people have, or you guys remember dermal wound healing, lecture exam 1? And how the thing, is gonna have a thick connective tissue scar? There are genes that shut off the fibroblasts. And if you have normal genes, you might have a little scar. Well, when people have keloids, that gene is defective, and the fibroblasts just keep making collagen, and you'll get this huge thing. This person has that defective gene, wanted to get an earring, and it started healing the piercing by forming this huge keloid.
Did it cut off?
Yeah, but guess what?
Keeps
Then it
going.
goes back. Because you still have a wound, and in the wound healing, the gene doesn't shut off. So you cut it off, then it goes back. And then you cut it off, and then it grows back. There's actually medications to hold it down though, if you know you're gonna form those.
My dad has, how does it, I was always wondering about how it's like called cauliflower ear, I think, from wrestling and stuff. How does that end up happening? Is it just from pushing down?
It's when you're accumulating scar tissue.
Just
Yeah. So this is both parts of the external ear auricle or pinna, and this is actually should be canal, external auditory canal. One thing that is not in lab is we have in your external auditory canal ceruminous glands. These are also modified sebaceous glands. They're forming something a little bit different from sebum, and it's cerumen or earwax. A lot of the bacteria that grows in the external auditory canal do not like cerumen. Some do, but most do not, so it holds down growth bacteria. But it's really sad when it gets caked like this. You're supposed to be able to see the tympanic membrane and even some of the ossicles on the other side. No, when it's caked with cerumen.
This from what, just not cleaning your ears?
Now some people just make more than others.
Oh.
And unless you know how to do it, you really shouldn't go try digging it out, because most people just pack it up against the tympanic membrane.
So what is the best way to clean your ears?
What's that?
What's the best way to clean your ears?
There's certain solutions that make this really soft, and you just gotta, and it's really inconvenient though, you just gotta sit with your head on a pillow so it just sits in your ear canal, and after so many minutes, then you turn on the other side and it just drains out.
My grandma used to use hydrogen peroxide, I think.
Yeah, it's not as effective as some of the other things for softening it up, but most of that action from hydrogen peroxide is effervescent, bubbling it off, instead of softening it. I swear to God, man, my kids, they would be so hard of hearing, and I call their names like multiple times, they'd never hear me, and I'd always tell them, hey, go lick a candle. Because my 3 kids seriously are serious candle makers. But I don't really make much earwax at all, but man, well, my ex did, and so all my kids got that gene. And then we went to go see, there those kids took them to see Shrek. And in the intro, Shrek was actually making candles with his earwax. Dad, they took your joke. Okay, tympanic membrane is the border between external ear and middle ear. For sound, for hearing, the sound goes from traveling through air to traveling through solid. There is actually an air-filled cavity on the other side of the tympanic membrane called a tympanic cavity. And in the tympanic cavity are 3 auditory ossicles. 3, 3, 3, and 3. Malleus, Inca, stapes. The foot plate of the stapes is a perfect fit for the oval window. On the other side of the oval window is the inner ear and perilymph. You guys remember perilymph from lab? So sound waves go from air, external ear, solid in middle ear, to liquid in inner ear. If you are hearing the sound of my voice, those sound waves are going through 3 different states of matter for you to be able to hear my voice. Air, solid, liquid. On this animation, I'm going to end it here. The function of the middle ear for hearing is to amplify the sound. And it actually amplifies the sound 22 times. The volume in your external ear to the volume in your inner ear. In your inner ear, it is 22 times louder. Okay? And the main reason why is because the size of the tympanic membrane is way bigger than the size of the oval window. This is the same amplification that you use with a stethoscope. You got the diaphragm that's this big, and you're putting that sound in that tiny little ear hole. Whenever you catch sound in a bigger space and put it in a smaller space, it amplifies it. This is 17 times bigger than this. Then just the physics of the 3 auditory ossicles also amplifies the sound slightly, like 1.7 times. 1.7 times 17 is 22 times amplification. Everybody got it? Okay, that's enough for today.